Welcome students. This module is from Unit 2 of the paper Microbiology and Plant Pathology. The name of the module is Bacterial Motility. Module number is 10. I am Dr. Janet Mascarenhas from Carmel College, Nuve. This module outlines the methods used for checking bacterial motility. In this module, we will be studying two methods, that is hanging drop method and the stab culture method. Learning outcomes. At the end of this module, the students will be able to explain the term bacterial motility, describe the hanging drop method and also describe the stab culture method for detecting bacterial motility. Now, what is bacterial motility? Bacterial motility is the intrinsic ability, that is the natural ability of the bacteria to move itself in the medium, that is it might be either solid or liquid medium in which it is suspended. Now, flagella is the organ of locomotion in bacteria. So, flagella are thread-like appendages which are extending out from the plasma membrane and the cell membrane of the bacteria. Now, there are various types of arrangement of flagella on the body of the bacteria, and this causes the variation in motility in the bacteria, as some are highly motile and others are slightly motile. Now, there are two methods to detect bacterial motility. First is the hanging drop method and the second is the stab culture method. Now, let us study about each one in detail. First, the hanging drop method. Now, this method is used for the microscopic observation of living microorganisms, especially bacterial motility is observed without staining them. Now, when we use the heat staining process, it will result in the death of the bacteria, which will cause a change in its morphology. Now, this method is a special type of bed mount in which a small drop of bacterial suspension is hung from the center of a cover slip into the cavity of a cavity slide. Then this hanging drop preparation is then observed under the microscope, preferably under reduced light, so as to enhance the visibility and for better contrast. Now, let us see how we have to proceed with the hanging drop preparation of the microbial culture. So, first we need to take a slide, that is a cavity slide which is having a concavity in the center of the slide. Then we have to apply either Vaseline or petroleum jelly around the concavity. Next, we need to take a square cover slip and put a drop of microbial culture onto the center of the cover slip using a inoculation loop. Then we need to apply petroleum jelly or Vaseline at the four corners of the cover slip. Then place the cavity slide over the cover slip in such a way that the cavity covers the microbial drop. Then we have to apply gentle pressure on the slide so that the petroleum jelly which is present will seal the cover slip and slide together to prevent the evaporation of the microbial culture. Then we need to invert the slide so that the cover slip is facing upwards. The microbial culture which was present on the cover slip will now be suspended in the cavity of the cavity slide. Next, this slide have to be observed under the microscope. So, when we see under the microscope, we will see that motile bacteria can be seen moving across the field of vision. Now, true motility can be seen as a rapid movement of bacteria across the field of vision, while the non-motile bacteria may show apparent motility due to the Brownian movement. Now, what is Brownian movement? It is the vibratory movement caused by the bombardment of the molecules of the liquid which will strike the cells and thereby causing its displacement. The next method is stab culture method. Now this is the indirect method for the macroscopic observation of bacterial motility. Now in this method, motility is demonstrated using a semi-solid agar medium. Now this semi-solid agar medium, it has a soft consistency. So this will allow the motile bacteria to migrate readily in the medium. In this procedure, the microbial culture is stabbed once into the center of the semi-solid agar tube with an inoculation needle up to a depth of half of the medium. Now care has to be taken while inoculating so that the needle has to be removed from the same place how it was inserted into the agar medium, that is the semi-solid agar medium. The cultures are then incubated at 37 degrees Celsius and examined daily for about 5 to 7 days. The bacterial motility is evident by a zone of growth which is extending out from the line of inoculation of the bacteria. The motile bacteria will spread out into the medium from the site of inoculation, while non-motile bacteria will remain at the site of inoculation. Now in this photograph, we can see there are 3 tubes. In the first tube, we have inoculated non-motile bacteria and incubated for 24 hours time period. In the second tube, we have inoculated motile bacteria and incubated for 24 hours. In the last tube, that is the third tube, we have again inoculated motile bacteria but allowed it to grow for a duration of 5 days. Remember, we are using semi-solid agar medium in these tubes. Now in the first tube, that is tube number 1, we can see that the non-motile bacteria has remained affixed to the needle tract, while in tube number 2, the motile bacteria has started migrating away from the needle tract. While in tube number 3, the motile bacteria has migrated throughout the tube in the semi-solid agar medium. So this proves, or in this tube we can observe the true motility of the bacteria. Now let us summarize what we have studied. Motile bacteria, they possess flagella which helps them in locomotion. Now there are 2 methods which are used for checking the bacterial motility. First is the hanging drop method. This is the direct method to check the bacterial motility. And second is the stab culture method, which is an indirect method. Now these are the references which I have used for this module. And these are some additional references, some websites which you can browse. Thank you.